Hi, everyone. Thank you for the opportunity to talk to you today. Please let me know, can you see my screen as a screen presentation mode?
Yes.
Okay, so you see the full screen, right?
Completely.
Oh, okay. So, just give me a second. Okay. So my name is Chimene Kesserwan. I'm a clinical cancer geneticist in the genetics branch at NCI. And before I joined NIH in 2020, I worked at St. Jude in the Division of Cancer Predisposition. So most of the work I am presenting today was completed while I was a faculty at St. Jude. I have nothing to disclose, and the opinions expressed in this talk are my own and do not reflect the view of the National Institute of Health, the Department of Health and Human Services, or the United States government. The first objective of the talk today is to review the scope of germline variants in childhood cancer. And the second objective is to demonstrate how the integration of matched tumor normal data could inform the classification and the interpretation of germline variants. First, I would like to set the stage by reviewing how the field of cancer genomics is evolving. The Cancer Genome Atlas and the International Cancer Genome Consortium have contributed significantly to our understanding of the landscape of cancer, specifically for adult onset tumors. In the pediatric space, large-scale analysis of pediatric cancer data are made available through the Pediatric Cancer Genome Project and the Target database. So led by Jim Downing from St. Jude and Elaine Mardi, then at Washington University of St. Louis, the Pediatric Cancer Genome Project started in 2010 and was completed in 2013. Whole genome sequencing was performed on 700 paired tumor normal samples. Whole exome sequencing and RNA-seq performed on about some 1,200 samples. So the PCGP expanded our knowledge of the molecular underpinning of cancer and led to a series of high impact publications. So the PCGP has also marked a new debut for the era of cancer predisposition in children. In a landmark paper by Zhang et al., 8.5% of children with cancer were found to carry a pathogenic or likely pathogenic germline variant in a cancer predisposing gene. The left figure shows category of the 565 cancer genes analyzed for germline mutations, as well as the list of 60 cancer predisposing genes associated with autosomal dominant inheritance. On the right, panel A and panel B show distribution of pathogenic and likely pathogenic germline mutations in different gene categories and cancer subtypes. Notice the enrichment of germline P53, which is largely contributed by a rich cohort of childhood adrenocortical carcinoma in the setting of Li-Fraumeni syndrome. So the TCGA and the PCGP and other discovery studies have expedited the understanding of molecular classification of tumors and the contribution of germline predisposition to cancer. The table here includes a list of the first clinical studies conducted in the US of matched tumor normal in children with cancer. My OncoSeq study from Ann Arbor included 102 patients with solid brain and liquid tumors. Cancer predisposition was reported in 10% of cases. Basic-3 study from Baylor included 150 patients with solid tumor, of which 10 patients harbor a PLP variants in a cancer predisposing gene. The Clinomics protocol from NCI analyzed 59 patients with high-risk solid tumor. 12% of this cohort harbor a germline predisposition to cancer. And PEP-seq from Columbia and New York examined tumor and germline samples on 101 patients with solid or liquid tumor. 14% of the cohort carry a pathogenic or likely pathogenic variant in a cancer predisposing gene. Meanwhile, St. Jude and leveraging on the Pediatric Cancer Genome Project findings, the Genomes for Kids Clinical Research Protocol was launched in 2015. The protocol is led by Kim Nicol, a national and international expert in the field of cancer genomics and predisposition. So every child enrolled on the protocol is offered to have his tumor interrogated by whole genome, whole exome, and transcriptome, and his germline interrogated by whole genome and whole exome. So the aim was to identify and characterize actionable diagnostic, predisposing, prognostic, and therapeutic variants. So in the study, 309 patients were enrolled. 85% had new cancer diagnoses and 15% had relapsed or refractory disease. No tumor was available for 47 patients, and 9 patients did not have sufficient tumors for analysis. 9 patients opted out of receiving germline testing. In summary, germline analysis was performed on 300 patients, and tumor analysis was done on 253 patients. Results from this protocol were recently published. I will be focusing on the germline findings only. So we identified 58 germline pathogenic or likely pathogenic variants affecting 29 genes in 55 patients. Therefore, 18% of patients carried a PLP. When I say PLP, just I mean pathogenic and likely pathogenic variant in a cancer predisposition genes. We noticed that 25 out of 58 of the PLP variants are in genes relevant to the patient tumor type. For example, retinoblastoma 1 variant, RB1 variant in a patient with retinoblastoma, whereas 33 of 58 PLP variants are in genes with no relevance or with uncertain relevance to the patient's tumor type, and or variants associated with autosomal recessive cancer predisposition syndrome, or with adult onset cancer, for example, a BRCA2 variant in a child with glioma. In addition, we detected 402 variant of uncertain significance, VUSs, affecting 111 genes in 230 patients. 77% of patients had at least one VUS. So the image on the right shows the list of genes with PLP variants in the G4K cohort and the distribution across cancer type. So questions we asked: can we use tumor data to better inform the classification of germline VUS? That was the first question. And the second question: can we use tumor data to better inform the relevance of germline pathogenic and likely pathogenic variants in respect to the patient's tumor type? This brings us to our next objective, which is to demonstrate how the integration of matched tumor normal data could inform the classification and the interpretation of germline variants. But first, I would like to briefly talk about the Mg Guideline, which were published in 2015. And these guidelines constitute a framework for variant classification that most labs have currently adopted. For the sake of time, since I don't have enough time now to go over it, I will just bring just key elements to this framework. So the evidence categories included in the curation process are represented here in this column and consist of population data, computational and predictive data, functional data, segregation data, de novo data, allelic data, and other. So each piece of evidence is assigned a direction, either benign or pathogenic, but also a strength like supporting, moderate, strong, and very strong. When we, for example, I will give you an example for pathogenic moderate, we use the population database. If the variant is absent from the population database, we assign PM2. P stands for pathogenic and M stands for moderate. So it's getting a pathogenic moderate weight as evidence. At the end, we after we have collected all the pieces of evidence, we combine them into a scoring rule, and we come up with a classification for the variant as pathogenic, P, LP, VUS, benign, and likely benign. However, The ACMG guidelines did not address how to integrate evidence generated by tumor data when assessing the pathogenicity of germline variant. Therefore, the ClinGen established a multidisciplinary working group, the germline somatic variant subcommittee, with this focus in mind. As shown in this graph, and after reviewing and analyzing available resources and performing somatic integration curation exercises, it was determined that somatic hotspot could be systematically integrated into a moderate evidence of pathogenicity, labeled green in this diagram. Just to make sure that we are all on the same page, somatic hotspots are variants recurrently seen in cancer. They are the drivers. So the second somatic data element that could inform germline curation is RNA-Seq data. If RNA-Seq is indicative of a splicing alteration, this may be considered as strong evidence in support of germline pathogenicity. Other somatic evidence, such as tumor mutational signatures, could be considered supporting evidence of pathogenicity. However, these evidence codes, labeled here in yellow, should only be assigned under guidance of a multidisciplinary tumor board. Also, this working group determined that at present, additional caution is needed to integrate focal loss of heterozygosity as an evidence to support pathogenicity to a germline variant. Yet, one question remains to be addressed: how tumor data may help to refine the interpretation, i.e. the relevance of germline variants relative to the patient's tumor in question. Germline pathogenic variants can be stratified as either relevant to the patient's tumor type or of uncertain relevance to the patient's tumor type. For example, if you have a patient with TP53 variant, this patient is a child and he has adrenocortical carcinoma, this pathogenic P53 variant is considered relevant to the tumor type. Whereas if you find let's say the same, we find a SMARCA4 pathogenic variant in a child with even adrenocortical carcinoma or neuroblastoma, then the relevance of this variant to the tumor type may not be that obvious. Similarly, the association of heterozygous BRCA2 pathogenic variant in childhood gliomas, for example, or other childhood cancer are not well established. So how tumor data may help to refine the relevance of pathogenic and likely pathogenic variant. So I will walk you, I don't think I will have time to walk you through the 4 cases. I think I may go over only 2 examples where integration of tumor data helped refining the classification and interpretation of variants. So all these examples are included in the Genome for Kids paper. So if we don't have time to go over this example, all of the example, you can refer back to the paper. But what types of tumor data we have interrogated? First, we looked at hotspot mutation. Then we looked at loss of heterozygosity or second mutational hit in the wild type allele. If present, this may indicate selective pressure caused by the mutant allele, which could serve as a supporting evidence of pathogenicity. We also examined tumor mutation burden and microsatellite instability as supporting evidence for mismatch repair deficiency. Now, for splice region variants of uncertain significance, we examined RNA-Seq data. Exon skipping or intron retention would be indicative of splice alteration. Finally, data from whole genome sequencing allowed us to investigate the mutational signature, especially for some canonical signatures, such as signature 3 and 8 suggestive of homologous repair deficiency, or signature 15 in mismatch repair, and signature 18 attributed to damage to the oxygen reactive species. So here is our first example. This is a 10-year-old male with B acute lymphoblastic leukemia and cafe au lait macules. He did not have any other findings, and the family history was not contributory. We performed the tumor normal next generation sequencing, and in the germline space, a germline variant in NF1 was reported as C.9621 plus 3A to G. The variant allele fraction was 60% in whole genome, 55% in whole exome. But using an alternate transcript, this variant is also designated as C.6858 plus 3A to G. Just to remind you, here are our canonical splice donor canonical splice acceptor. The canonical splice are in general plus minus 1 or 2. Our variant is +3, which is 3 nucleotide upstream of the preceding exon. So it's not considered a canonical. So following the ACMG guideline, this variant is classified as VUS. Criteria meeting and evaluation strength that we applied for this variant are in silico prediction data and population data. Using Splice AI, this is a publicly available Splice prediction tool that we adopted in ClinGen. This variant is predicted to cause splice alteration via donor loss, as you see here. So this evidence was assigned a supporting code PP3. The next evidence that we examined is the presence or absence from population database. So this variant is absent from NOMAD. Therefore, we applied the PM2 supporting evidence. We could not find additional evidence criteria such as functional studies, which have a higher weight, and we did not have segregation data for this patient, or even in the literature, we did not find segregation data for this variant that would help inform the classification. So with only the supporting evidence and without integrating the somatic data, the variant was classified as variant of uncertain significance. So here are our somatic findings. We have NF1 intragenic deletion. In addition to the one that we found in the germline, now we have NF1 intragenic deletion spanning exons 3 to 8. So this constitutes a focal loss of heterozygosity. And we know that focal loss of heterozygosity, as I showed you in this graph, supports pathogenicity of truncating or null variant additionally, in addition to the focal loss of heterozygosity, we have Rna-Seq data. The data was indicative of exon skipping. So the image here on the right display a histogram showing Rna-Seq coverage relative to the NF1 gene. Canonical splices are shown as light blue links between exons, and a non-canonical splice is shown in mauve. So the height of the mauve and blue lollipops is proportional to the number of splice junction reads detected and plotted on a log scale on the y-axis. The purple bar indicates the position of the NF1 exon 45 splice donor mutation. So you see exon 45 clearly is expressed at a lower level relative to the flanking exons. And a non-canonical splice linking exon 45 to and 46 is observed, indicating, in fact, an exon skipping event. So according to the ClinGen germline somatic integration diagram, PVS1 for pathogenic very strong code may be considered under guidance of tumor board. Now, with the integration of somatic RNA-seq data, then this NF1 variant was upgraded from variant of uncertain significance to likely pathogenic, given we have now 1 strong and 2 supporting evidence. What about relevance to the patient's tumor type? In terms of relevance, given that the germline variant is associated with focal loss of heterozygosity, is suggestive of selective pressure and is indicative that this variant is likely relevant to the patient tumor. Now moving to case 2. This is a 16-year-old female, was diagnosed with Ewing sarcoma of the pancreas. Per tumor normal performed germline whole exome sequencing showed a BAP1 variant designated as C.256-3C2A. This is a splice region alteration in which AC is replaced by an A in intron 4, 3 nucleotides upstream of exon 5 of the BAP1 gene. Now we have an acceptor in the splice region, in the acceptor region, but it's not a canonical splice again. We first classified this variant without integration of somatic data. So the variant is predicted by Splice AI to cause splice alteration with a score of 0.23. Therefore, we assigned a PP3 code for this evidence. And this variant has a very low minor allele frequency in NOMAD, which allowed us to assign PM2 supporting code. With only 2 supporting evidence, the variant is classified as VUS. Now we will examine the somatic data. So the tumor, as we expect, because this is Ewing sarcoma, as I mentioned, showed EWSR1 fly fusion, translocation and fusion. RNA-seq showed intron retention. So this alteration causes read-through between exon 4 and 5 with intron retention shown as high level of read coverage in the intronic region. In comparison, RNA-seq from 3 genome for kids Ewing sarcoma cases did not show increased coverage of intron 4. So the inset histogram here shows a relative proportion of wild type and variant alleles in tumor whole genome sequencing and RNA-seq. So based on RNA-seq and according to the ClinGen germline somatic integration diagram, a pathogenic very strong code, PVS1 code, what may be considered under guidance of institutional tumor board. Therefore, after integration of somatic data, the variant was upgraded from VUS to likely pathogenic. Now, in terms of relevance to the patient's tumor type, given that the wild type allele is not lost, we think that this variant is likely not relevant to the patient Ewing sarcoma. It could be like it's an incidental variant, a secondary finding, not primary finding contributing, contributing to the tumor. Briefly, I will summarize the third case. So our third case is a 5-year-old with ganglioneuroblastoma. Germline testing showed a missense variant consisting of substitution of alanine to threonine at codon 161 of the TP53. Based on the Mg guideline, this variant is classified as VUS. So criteria meeting an evaluation strength are shown in this table and include functional evidence. Here we applied functional evidence because we have a TP53 variant curation expert panel where they specified rules for the P53 and what are able to, of course, we have the IARC database to support with a lot of supporting functional data to query. So criteria meeting an evaluation strength are shown in this table and include functional evidence, population data, and in silico evidence. Next, we wanted to ask the question whether this variant is a mutational hotspot. For this, the ClinGen Germline Somatic Working Group recommends to query a publicly available database maintained by the Memorial Sloan Kettering Cancer Center called cancerhotspots.org. Determination of hotspots was based on a mathematical model to identify driver mutation in about 24,000 tumor samples. So in this website, A161T is a somatic hotspot seen in 12 samples. Therefore, a moderate evidence of pathogenicity can be assigned per recommendation of the ClinGen Germline Somatic Working Group. So the TP53 VSEP, we know that this is a hotspot, and we adopted this code just to mention that the TP53 variant curation expert panel adopted PM1 for hotspot evidence, and we applied this rule as well. Therefore, adding PM1 code to A161T will lead to variant upgrade from VUS to likely pathogenic. Some additional relevant somatic data for this variant include the demonstration of a monoallelic expression of A161T on RNA-seq. So 6 markers, including the germline variant C2T, are represented by 3 rows of vertical bars. The bars are colored red and blue to show the allele fractions. The rows indicating germline and tumor DNA show most positions as heterozygous with both red and blue portions. However, in the RNA, all markers are monocolored, indicating that only one allele is expressed. So at the location of the germline variant, the G2A, the bar is completely red, indicating that only the variant allele is expressed. So we have allele-specific expression, suggesting that the wild-type allele is transcriptionally silenced. Therefore, we think that the P53 A161T is likely relevant to the patient's tumor. I will not have time to go over the last example, but to bring to your attention the fact that a summary of all pathogenic variants with corresponding tumor data and relevance to tumor types are included in this table. These examples demonstrate the power of 3 platform sequencing to refine classification, and interpretation of germline variant in cancer predisposing gene. So as take-home points, simultaneous assessment of tumor and germline genomes, hotspots mutations can improve variant classification. However, at present, other somatic data are not recommended to be systematically integrated. Tumor RNA-seq and tumor phenotype should only be considered under guidance of multidisciplinary tumor board, as this requires patient-specific integrated data and institutional norms. Additional caution needed with LOH to ensure focal loss and relevant germline variant on retained exon. It was wonderful to be part of the Genome for KIP team at St. Jude and to work with many talented people and amazing genetic counselors. Special thanks for my former boss and great colleague, Kim Nichols, who is a PI on this protocol. I would also like to thank the ClinGen Somatic Germline Working Group team and the great leadership of Sharon Plan, Dimitri Sankin, Michael Walsh, and Deb Ritter. And with that, thank you so much for listening, and I will stop sharing.